Hello, and welcome to today's webinar, Translating Insights into Better Outcomes, announcing the new QIAseq Human Exome integrated solution. My name is Brian Dugan. I'm an Associate Director of Global Product Management for the Genomics Group at QIAGEN. Over the last few years, QIAGEN, which is obviously well known in the sample prep side and our Over the last few years, QIAGEN, which is known for its sample prep solutions and for its bioinformatics solutions, has built a powerful next-gen sequencing portfolio. The next-gen sequencing portfolio that QIAGEN has built has been largely on amplicon-based technologies, specifically the underpinning technologies, things like single primer extension, which allows us to do one primer at a time as a target across the genome, chemistries that are robust for enriching in multiple different facets and multiple different analytes, including DNA, RNA, methylation, and more, as well as adding things like unique molecular indices and unique dual indices. And so all in all, QIAGEN has been able to put together a comprehensive solution for many researchers to be able to look at all of these different analytes with this technology. But as we move on to the future, we know that there's a need to be able to essentially be able to do large-scale genomics. To do that, QIAGEN is announcing our new QIAseq Exome solution. And what we're really bringing here is now the addition of two different things into this technology, into our QIAseq portfolio. The first is the addition of hybrid capture as a way to enrich for the genome. And the second is really pairing the Workbench side with the in silico side to bring about integrated solutions. So now we're single primer extension and primer-based solutions. We're really focused on small panels or even medium-sized panels. Now we're able to do genome-wide or ultra-large panels that allow us to look at things not just from one specific area or a small area of the genome, but really expanding onto many megabases and covering a tremendous amount of real estate and be able to look at things not just in cancer, but look at focused inherited panels and really be able to see all of the exons simultaneously. Now, part of this is driven, right, because we know that there's a number of trends that are going on in the market and overall. The cost of sequencing is rapidly decreasing. And in fact, just this year, it even decreased even more as the price of some of the NovaSeq flow cells has decreased considerably. This drop of pricing and this now access, greater democratization to sequencing capacity allows larger and more complex experiments to be done. And so with the fact that now there are more high throughput sequencers than ever before, that costs are lower than they've ever been before, it now makes exome an even more, a bigger opportunity here to be able to look at that and to do more experiments than ever before using a genome-wide panel. But one of the challenges here with doing exome sequencing is not the amount of data that you can generate, but the fact that the variants and the interpretation of those variants is still a clear and present challenge and a bottleneck that we will discuss later on in the presentation. Now, as I announce our QIAseq Human Exome, it's a true solution to detect pathogenic variants with confidence, ease, and efficiency. And there are three main points here to really start off discussing. The first is that while getting this comprehensive detection of disease-causing variants, we actually have a solution that will help reduce your sequencing cost per sample by up to 50%. Our solution is a single-day, automation-compatible workflow that is specifically built with scale in mind. It's also built with the ability to go from essentially any type of sample as well, and we'll talk about the differences between using blood samples, tissue samples, and circulating cell-free DNA. And finally, as we go to the second part of the presentation, we'll talk about the way that the Workbench side integrates seamlessly into the bioinformatics side, allowing you to take a variant file and go through a comprehensive interpretation to reduce your overall turnaround time from sample to insight. So first, let's talk about the first point. So one of the biggest challenges when doing exome sequencing, and I talked about it previously in the current trends, it's not so much the cost of the panels themselves as it is the cost of sequencing. And when you look at the cost of sequencing, you can see that it varies tremendously. Right now, up until recently, a whole human genome cost about a little under $1,000 in sequencing reagents, and that's dropped by about 75%. But exome sequencing itself and the ability to sequence deeper than you would with the genome, right, has always been considerably cheaper. But the cost is really associated with two different things, specifically with how large the size of the panel is and how efficient is the panel. And so looking at our QIAseq Human Exome, and just a brief discussion here, is here you look at our panel here, our QIAseq Human Exome, which essentially with 95% of the sequencing at 30x generates about 8 gigabases of data. When we look at two competing suppliers, you see that the amount of sequencing data to get that same coverage is at 10 gigabases and 15 gigabases. All of this with higher fold and 80 penalties. So all in all, what you see is that all of these things together lead to your ability to actually multiplex more on a QIAseq Human Exome panel with less sequencing cost. Now I'll show you about how this results and how this actually impacts variant detection in some following slides. But you can see here that just on a NextSeq 2x150 run, that you can do almost 40 to up to 50% more samples, right, at a single run. This gives you the flexibility to take either more samples and to sequence them simultaneously, or to sequence deeper on some precious samples. So let's talk about the kit. So the kit, as I said, is a single-day workflow. It's automation compatible, and it really allows you to go from sample to data. The first step, as in many exome workflows, is doing library preparation. So here we show our QIAseq FXDNA library prep kit. The QIAseq FXDNA library prep kit is actually a enzymatic-based library prep that allows you to do library prep without any type of mechanical shearing that can take under 3.5 hours. All of this is currently automatable on multiple platforms. The next step in this is now the enrichment, the QIAseq Human Exome Kit. Now the advantages here is that there's a number of different hybridizations and times that you can actually take, really based on what you're looking for. With the hybridizations that can be very, very quick to overnight ones, you have flexibility to essentially to slot into your current workflow or be able to speed it up into what might be some of the fastest solutions on the market. And after that, it's simple sequencing on the Illumina platform, and then analysis using our QIAGEN CLC Genomics Workbench and the QIAGEN Clinical Insights solution. But as I said in the previous slide, that while we show the QIAseq FXDNA library prep kit, which is enzymatic fragmentation based, many customers have different preferences on how they choose exome sequencing. And so to be able to meet the demands of many of these different types of customers, we have other kits as well that are compatible with different types of approaches or different types of samples. The QIAseq Ultralow Input Library Prep Kit is a solution that allows you to use existing covirus workflows that's flexible from a single nanogram up to 500 nanograms of input, and all while using this physical shearing method. Now, the other solution now in looking at circulating cell-free DNA is our QIAseq Cfdna library prep kit. This actually adds the QIAseq Ultralow Input Kit plus our QIAMP circulating nucleic acid kit. So essentially, all you need to do is have your tube full with blood, and then it's an isolation and a library enrichment in a single box. Again, this is compatible with covirus or physical shearing methods. right? And it is a very flexible solution to get you right into enrichment. But as I said, the workflows for hybridization as we move into that step are quite flexible. Now, there are many different types of hybridizations that are possible with the QIAseq exome kit. The first is as quick as a 30-minute hybridization. The longest is a typical overnight hybridization, right? And there are times in between from 90 minutes to 2 hours to 4 hours. But what I'm showing here on the right side is a figure that shows the targeted bases and the X-fold of mean coverage. And what you see here is that, without a doubt, his hybridization time will have an effect on how the representation of bases happens. But when you look at a comparison between a 4-hour hybridization and an overnight hybridization specifically, you can see there's not a significant difference in terms of performance. Now, what this means is this allows you to still do a single-day workflow without having to do an entire overnight hybridization, which can speed up your solutions here. Without the overnight hybridization, you can simply on the same day go right into your sequencing reaction and onto the sequencer and actually start that up to 8 hours faster. Now, there's different types of hybridizations, and if you need to do things ultra fast, you can always go down to 30 minutes worth of hybridization as well. So all in all, it's a highly flexible solution to meet any customer need. Now, in terms of performance here, right, when we talk about the fact that this kit really provides uncompromised performance, and is equivalent performance on target coverage regardless of whether it's a single or multiplex hybridization and capture. So there are different solutions out there to allow you to do a single library per pool or multiple libraries per pool. But what you see here on the figure on the right, again, looking at X-fold of mean coverage, there's essentially no difference between doing a single library in a pool and 8 libraries in a pool. All in all, this really just shows that these pools are highly efficient in ligating and pulling down on the libraries. Now, the other thing to think about, and I brought up Cfdna earlier, is that Cfdna can be quite challenging. And this is specifically because DNA in serum and plasma is degraded. And so here we took a look as a comparison between looking at high-quality genomic DNA and different FFPEs. I'm sorry, talking about FFPE, and I'll get into Cfdna in the next slide. But here looking at FFPE and the different qualities. So here in the comparison between genomic DNA and FFPE DINs of 3.5, 4.4, and 4.7, obviously the high-quality genomic DNA is going to provide you the highest quality in terms of representation of what you get from your targeted bases. But when we look at more samples that are more degraded, right, from as low as 20 nanograms here, right, starting from this lowest level of the 3.5 DIN, is that you can see that right below that dark blue line is that all 3 DINs really perform essentially simultaneously, really just overall. So you don't have a tremendous amount of drop-off, even from looking at high-quality DNA or different types of quality from FFPE. All in all, right, this shows you is that the library prep plus the enrichment solution will offer you a robust solution for the most challenging samples. Now here is the slide discussing Cfdna. And again, Cfdna is similar in terms of FFPE, maybe perhaps not having as much degradation, but certainly fragment sizes of DNA are much smaller than high molecular weight DNA that you would derive from cells. But you can see here again, when doing a comparison of 50 nanograms of high-quality genomic DNA to either 10 nanograms or 20 nanograms of Cfdna, that there's essentially no difference between either one of those Cfdna plots. Now again here, it's just only slightly less performs as well than genomic DNA. But again, the enrichment that you will have and the amount of coverage, the average coverage that you will have across the targeted bases is essentially commensurate. And so if you look at 10 nanograms and 20 nanograms as well, right on the left side now, if I look up to 40x coverage, I see 90+% of the target bases are covered at a minimum of 40x if I sequence to about 80x average in terms of the exome. So this again shows that the quality of the total solution will deliver you high quality sequencing results from some of the most challenging samples. Now, another aspect here, which is often important to look at, is representation or base representation. Now, typically, genomes themselves should be balanced in terms of GC and AT content, but at times you can tell that certain solutions, whether it's in the library prep or the probes, may have certain sort of biases towards certain compositions here. And you can look at the percentage of GC as a real good indicator of this. And what we're showing here with the QIAseq Human Exome is that if you look at a midpoint here of this graph, the percentage of GC of the target, is that you want to come close in terms of this heat map of right in the middle, right, about 50% as the genome is. But when you look at comparing solutions that are on the market, you can see that representation can rise and fall, really significantly, much farther than 50%. So again, coming back to the performance of the QIAseq Human Exome, our exome performs in a really balanced way, giving you equal representation across the genome, which will lead to higher quality calls, better representation, and lower amounts of dropout in regions of the genome. Now, to further illustrate this, we chose a gene, ADAT3, and we looked at this because of its high GC content. It's specifically challenging to sequence because it has approximately 65 to 70% GC content. And here's a comparison on the right side of two different pileups. One on the top is the QIAseq Human Exome, and the other is from another supplier. Now, when you look at this, just visually on the right side, you can see that both sets essentially cover this entire gene. But the reality is that pulling this down is not really possible, or is more possible with the QIAseq Human Exome. You can see here from the lines is that there's total coverage across this entire gene, even despite the fact that this extremely high GC content. And so now this was done with an average coverage of about 100x coverage. And 100% of the bases of this targeted region are actually covered. And it's actually covered at an extremely high rate, about 89.3x coverage is overall covered. So when you think about this, and going back to the representation and the importance of this leads you, again, to just further proof that with our solution, from library prep to our probes, and the total solution with that, is that you get an efficient solution to deliver high quality, high representative data that will allow you to make the best calls. Now, what's often being asked now of exome sequencing is not just calling SNVs and being able to call structural variants such as copy number variation. And the QIAseq solution actually works seamlessly with our QIAGEN CLC Genomics Workbench that has a pre-configured workflow directly in there. And so what we wanted to do was show our ability to do CNV detection using two known standards, Horizon Reference Standard, DNA HD753, that has known CNVs in MET, and then MIC-N. And what you can see here is that you can essentially, the probes are designed to be able to cover this region, right? But this analysis allows you now to be able to go through and to specifically look at, from the read pile up and look at the probes, to be able to tell if there is a copy number variation. So again, it's not just calling single variants, right, or single nucleotide variants, but also doing structural variants such as CNVs. Now, what is, exome sequencing now has been used for almost a decade. From 10 years ago, the first probe sets that were released on the first technologies, on the Illumina technologies and solid technologies, right? And it has evolved. And over that time, what we have seen is certainly changes in standards and things that need to be called and be reported. The first of those to be called out is obviously the Acmg 59, right? Genes that are specifically important to be covered, and that Acmg really has put a specific emphasis on. And so in doing an analysis of how we perform in these genes, we did a number of exomes at 100x average coverages. And what I'm showing you here on the left is how each of the base coverages at 100x are covered from a percentage point of view. So on the left side, you see total bases that are covered. At 1x, the total bases that are covered in all of the Acmg 59 genes is 99.66% of the bases. If I go all the way up to 40x coverage, 96.3% of the bases are covered. And so again, whether you're at 1x or 40x, it makes a significant difference in your ability to call variants, right? 40x, especially for many of these genes which are associated with inherited disease, 40x is sufficient to call many different variants. And so to have that high percentage now, again, leads to higher quality results in variant calls. On the right side, though, the other thing to consider is end-to-end coverage. It's not just single bases here. And while you're not going to have a significant amount of dropout, right, if you're covering 99 or 96%, it's also important to understand from structural variation analysis how you're able to do from an end-to-end basis. The table on the right side here, right, is showing this. So again, at 100x average coverage on the exome, we looked at what is the end-to-end coverage of each of these Acmg 59 at 20x. And at 20x, 98.2% of the bases are covered continuously. There's no breaks in it. And a single base pair break, right, is all that it essentially takes to drop that out. But again here, also looking at 40x coverage, 95% of the bases are completely continuous. So again, the reads are unbroken, the coverage is unbroken, and delivering you those high quality results. But the question always comes back, well, how does this actually affect things when I go to call variants? And so when we look at the QIAseq exome and its workflow with both our CLC Genomics Workbench and pairing it with our QIAGEN Clinical Insights, we looked at the HGMD database, and specifically looking at the size of our panel compared to other providers and our ability to detect known HGMD disease mutations. Now, going back to the value that I talked about earlier here, you can see here that the QIAseq Human Exome is smaller than other providers that are out there by a few megabases, you know, really at a minimum. But in spite of that, the QIAseq Human Exome actually will cover and pull down and detect more disease-causing mutations in HGMD, even given its smaller size. And when you compare that to other solutions which have larger sizes and will cost more to run, right, the fact that you can still, with a more efficient panel, pull down more mutations gives you a higher confidence in the fact that the areas that are most relevant to human disease are being covered, pulled down, and actually analyzed. Because it's not just about the probes, it's not just about the sequencing, it's also about the analysis that goes along with it. It shows clearly that we don't also miss less of the HGMD disease variants that are out there as well. And so again, you know, looking at the combined value of higher calling of HGMD variants and lower costs, this is a highly efficient solution that you can implement in your lab. Now, one of the other things to consider here is that when looking at exome sequencing, you know, to look at HGMG at a variant level is certainly something that many people do. But so much of exome sequencing is being done in a way where it impacts disease, and it impacts, you know, people who potentially have this disease and research into these diseases. And so with looking at HGMD variants, what we did is did an analysis and putting these into buckets based on the diseases that are known, and just a small subgroup of some of them. And so we associated it with cancer-associated diseases, inherited cancer-associated diseases, specifically things like hearing loss, cystic fibrosis, Tay-Sachs, Marfan, and then OI as well. And what you have here is that even compared to other solutions that are out there on the market, the increased detection of the HGMD variants is important, but when it is directly associated with diseases and linked to those things, you're actually going to find more. And I bring this because essentially this is why exome sequencing is really able to be done, right? It's for biomarker discovery, or it's also to be able to look at things at a genomic level for a broad basis to understand potential some diseases. And there really is no better solution that's out there, even despite the smaller size compared to other solutions, to be able to pull down these important variants. So all in all, right, with the size, the efficiency, the automatable solution, and being able to deliver insights here in some of these key areas, you can see this is a true end-to-end, highly valuable solution. But as we move into the next part, right, and I'll briefly touch on this, is that we know that the bottleneck here is not just on the front end, but is everything that I've talked about with data analysis and variant interpretation. And so we're about to move into our second part of the presentation where Rupert Yip, also from QIAGEN, from our QIAGEN Digital Insights team, will walk you through how you go through the FASTQ files after they're generated, and go through our analysis with our QIAGEN CLC Genomics Workbench, all the way through QIAGEN Clinical Insights for QIASeq. But as I close up and I transition over to Rupert's time, here I just, again, talk about the main bullet points. First, the new QIASeq Human Exome is a comprehensive solution for detection of disease-causing variants with potentially up to 50% reduction in sequencing cost per sample. It's a robust, single-day, automation-compatible workflow that will fit almost all of your needs. Do you prefer non-mechanical shearing versus mechanical shearing? Do you prefer to have it automated or not automated? Right? All of these things are capable with our different QIASeq solutions. And now, Rupert, I'll leave it to Rupert to talk about our rapid data analysis solutions and how we're able to deliver, really a best-in-class end-to-end sample-to-insight solution for doing human exomes. I want to thank you for your time, and I'll now pass it off to Rupert.
Thanks, Brian, for that excellent overview of the QIASeq exome. What I'm going to talk about now is how to take that high-quality data that is coming off of the exome operation and how one would analyze and interpret the detected variants coming off of those exome samples. So as Brian had alluded, some of the challenges with analyzing an exome has to do with the volume of data that comes off of an exome. We're talking somewhere between 50,000 to hundreds of thousands of variants that you need to sift through. As well, some of the callers, the algorithms that are used to detect variants are not accurately detecting some of those variants. And sometimes it requires multiple analysis types to detect relevant mutations. As well, with interpreting human exomes, once you've identified candidate variants, you need to refer to multiple annotation databases to get some information about those detected variants. And then, of course, you don't want to manually do the due diligence on all 100,000, so you want to have a reliable way to sift down to only the interesting variants to say more. And finally, once you do identify those variants of interest, you need to look them up in all those different databases, read the publications, and adhere to society guidelines on how to properly interpret those mutations. So today I'll talk about the analysis and interpretation portion of that workflow. That includes things such as variant triage, variant annotation, and I'll talk a bit about some of the challenges with computational speed and accuracy of variant calling, as well as the pipeline output. And then finally, once you've identified those variants, how to annotate, triage those variants, some of the algorithms that are used to do the expert curated content that's associated with those annotations, and as well as how to prioritize those variants. So first, I'll talk about the analysis, which is the detection of the mutations. This is primarily the workflow that comes off of the sequencers where you've got the raw FASTQ files. And our solution allows you to then send those FASTQ files to a cloud-based analysis platform. Some of our customers have their own bioinformatics team and has a homebrew bioinformatics pipeline to generate file. There are, of course, commercial off-the-shelf solutions that are available, and then, of course, QIAGEN has its own solution called the QIAGEN CLC solution. And all of them, as long as they generate a BCF with version 4.0 or above, will be compatible with our interpretation solution. So in the past, these pipelines to generate the BCF file has been quite cumbersome and required IT knowledge, where you have to perform that analysis through command line. The QIAGEN CLC solution presents an elegant, user-friendly interface that allows a biologist, really, with minimal IT training to be able to kick off these types of analysis. And one of the beauties of the QIAGEN solution, it is purpose-built for the QIAseq Exome, wherein we have a specific plugin designed explicitly for the QIAseq Exome. So you can see here that in the one of the plugins that you can select in the CLC Workbench is the QIAseq Panel plugin. And with that plugin, it allows you to, with a wizard, allows you to walk through the setup of how you want to configure and parameterize the analysis of your QIAseq Exome data. Once you've analyzed that data, we have a variety of tools that allow you to quickly review the results and pilots, including a genome browser. More importantly, we have powerful visuals metrics in order to QA and quality control the data that comes off of the sequencer so that you ensure high quality data is used in the final interpretation results. Next, I'll talk about the interpretation workflow, and that includes the variant triaging interpretation. What exactly is variant triage? Well, if you're working with exomes, and particularly with genomes, where you could have anywhere up to millions of mutations, obviously you don't want to do the manual curation of all millions. You want to try and sift through out of those millions down to the tens or the handfuls that are likely causing the phenotype or disease of interest. And so the ways people typically narrow down the variants of interest is through a series of selection criteria or filter criteria, where you remove the common variants out of the raw data, and of those, remove those that have no obvious effect on protein. And then of those, remove those that are not in the correct context of the disease that you're looking at. So for example, if you're looking for a homozygous or a heterozygous type of mutation, or if you're looking for a de novo or a particular mode of inheritance type of mutation, that allows you to sort of iterate on your hypothesis of the genotype of the disease of interest. And then based on those remaining variants, you might want to say those remaining variants show me those that have any relationship, direct or indirect, with the disease or the phenotype that I'm presented with. So those are the ones that you want to do the deep dive and manual curation on. And so having this variant triage workflow allows you to quickly go from millions down to the tens. So the traditional method of manual curation is fairly, you might start off with the raw VCF file that has, like I said, up to 1 million variants. You may even have a trio of them if you're doing a trio analysis. And from there, you might want to have additional metadata such as phenotype, pedigree information to describe the relationship between those various samples. And then from there, you want to identify if there are like cases that are found in databases such as ClinVar, and whether you want to make sure that your variant curation follows society guidelines such as Acmg. From there, you might want to see whether the mutation of interest has a certain rarity among a population of healthy individuals. So you go to databases such as Nomad, Genomes, and then you might want to look up additional annotations in the browser. From there, you might want to look up in silico predictors, so you'll use tools such as Mutation Taster, AD, and then finally, when you do have mutations that satisfy all of your criteria, you might want to go ahead and go into PubMed to look up those papers, and then ultimately, or even Google Scholar, and then finally going into reading those papers. So it's a fairly tedious process. With QCI Translational, we automate that process. Again, you start off with those various BCF files, and you have the metadata. You can combine that into the platform, and the platform will do the variant triaging to quickly go from the millions to the handful of tens. That information is fed into the knowledge base where you can ask those relationships, direct or indirect, between the remaining variants and the disease or phenotype of interest, to quickly generate a list of annotated variants with relevant and then finally you can export the annotated variants into a text file for follow-up reporting. So we get our knowledge into the knowledge base from several sources. They come from public databases, from licensed databases, as well as QIAGEN proprietary databases. We then apply that knowledge across society guidelines so that it's computed in a consistent fashion, as well as applying in silico predictors. And so our content is really updated on a basis. The guidelines are refreshed on a quarterly basis. We have we draw from over 40 different database sources, and we have been doing this for over 20 years, where we employ over 200 MDs and PhDs who do nothing but read thousands of articles a month and manually enter that information. The QCI platform really is a platform that allows the streamlined aggregation of samples. So you can do sample inventory and management of those samples on the platform. It allows you to do that variant triage prioritization that I mentioned. Once it's prioritized those variants, it then computes variant classification within the context of the phenotype and disease of interest, and then combines the analytical tools with the knowledge that's generated. So with that, I'm just going to quickly dive into a quick demo to see how all of those workflows are executed in the application. So once you log into QCI, QIAGEN Clinical Insights Interpret, you're presented with the list of analysis that you've created. And to look at an analysis, you simply select any of these. To upload samples, you go to the sample list page that shows all the samples you've ever uploaded, as well as the sample management inventory here. So to upload samples, fairly straightforward. You click Upload Sample, and then you can upload sample. So I'm going to go and select a QIAseq Exome sample here. Whoops, let me just move this out of the way. And then just submit, and it uploads the QIAseq into our servers. It does a validation to make sure that that BCF file is compliant. Parse it. And once that sample is up there, you can go ahead and create an analysis with that sample. We'll go ahead and select a previously created filter setting. And then select uploaded sample. I can click and drag that in there. If I have other sample metadata, add that here. Or I can continue. If I have clinical symptoms, I can go ahead and enter that here as well. I'm just going to go ahead and edit. I'll go ahead and then to take a look at the analysis, I've already pre-created one here. We can go ahead and take a look at how the annotations work. So here you have an overview. This is what we call the variant list table. These are the variants that are the list of prioritized variants from a starting list of over 38,000 variants, and we were able to filter down to 100 variants. And of those 100, we prioritize based on pathogenicity. To understand what filters were used, and you can click on the pencil icon here to open up, and you can see that we filtered. The confidence filter is really looking for high-quality variants, variants that are, for example, a Phred score of 20 or greater. I didn't select this, but if I selected this, a minimum read depth, genotype quality, and so forth. You can also say, I'm looking at variants with a rarity of less than 1. Adjust this here if I wanted to. And then the biological context filter really is looking for any disease, any variants of the 2,264 that are related to hearing loss in this particular example. But I could have selected any biological experiment. If I want to add additional filters, I click the Add Filter to add more. So, for example, the Predicted Deleterious filter allows me to select variants that are computed as pathogenic, like according to Acmg guidelines, or are listed within the HGMD, ClinVar, or CentoMD database, or are associated with gain of function or loss of function. So those are the various filters that you can use to quickly go down from the hundreds of thousands down to the tens if you want. To do a deep dive on a particular variant, so this has already prioritized these variants based on pathogenicity. I can double click to go into this to explore this variant a little more. So this is the deeper dive that I'm talking about. And the system had computed this variant as pathogenic. And here are the criteria that were triggered, the Acmg criteria that were triggered. There was about 5 criteria that were triggered. And the evidence to support the triggering of the PS4 is listed here. So I can go in and take a look at the evidence by clicking on it. And it showed that there were 4 papers that triggered that evidence. PS4 really is identifying any cases that were consistent with this mutation and this disease. And so you can see that there's this paper published this year that describes this particular mutation, the L1160 deletion, associated with congenital hypothyroidism, for example. And so you can read that paper, or you can read sort of like a quick summary of what this the relationship that is. If you believe that paper and you believe the evidence, you can just leave it alone. But if you don't believe the evidence, you can X this out, and it will recompute the classification based on the absence of that evidence. You can then, based on your own expertise, you may decide that this is, maybe more of a likely pathogenic versus a likely pathogenic, depending on how your lab likes to interpret these, and then set the assessment. But you always have this evidence that is clearly transparent how we computed last. The variant details pane really more details about the variant. There's if you're not familiar with the gene, you can click on the Gene Reviews of the Genetics Home Reference link. If you have that particular BAM file open in IGV, you can click on this to open up the genome browser within IGV to view the pileups, or you can just go back to the CLC tool pileups if you want. The effect on protein is fairly useful. It allows you to see where in the gene this mutation exists. So in this particular example, it exists in this, so each rectangle represents an exon, so it seems to be showing up in this particular exon of the gene. This yellow area represents the functional domain of that protein, and it shows that this mutation occurs in the functional domain of the protein. And these red dots represent other pathogenic knowledge base. Clinical cases from other laboratories can be found in the clinical cases section. And so in this particular example, there is one example of this mutation found in the HGMD database. The rarity in general population allows you to look at the frequency of this particular allele in a general population. So you can refer to the allele frequency community, NOMAD database, or the exon variant server information here, as well as 1,000 genomes. Reported functional impact really looks at in silico predictions of the impact of this particular mutation, and so you can look up CAD scores, mutation taster, max nth for splicing, as well as gene splicer, BSIF as well. And yet again, another genome browser option, which is if you have BAM and text files available, you can point to those and look at it. There's also the bibliography of this particular mutation if you want to read all the papers that are associated with this mutation. There's 24 papers or references associated with this mutation, but if you're only interested in clinical cases, you can just click and see the clinical cases, or just filter for only functional studies. So for this makes it very zero in reference. So that's fairly straightforward in how you sort of upload and create an analysis using QI. So now to finish up, in summary, really the QCI solution for the QIAseq exome is designed to streamline that interpretation, going quickly from the hundreds of thousands down to the handful, so that you can focus your effort on only those handful. And it's really a solution designed to address those challenges of significant amount of data that's generated from sequencing effort. And it standardizes the way in which you can interpret these variants, because we adhere to the Acmg criteria guidelines, and allows you, while I've shown an example of just a singleton, you can certainly do trio analysis with this platform, as well as case cohort analysis. With these tools, it allows you to quickly do that manual curation, that due diligence, to look across multiple annotations from various databases to quickly understand the effect of the impact of that mutation on this sample. It allows you to identify the relationships between the disease and the gene, or even variant interest. And it quickly allows you to scan across various databases, standardized across guidelines. And with that, I conclude this portion of the presentation, and open it up to questions.